The first phyla of fungi is phylum Basidiomycota, which is also called club fungi. This phyla gets its name from the reproductive structure called basidium, which resembles a club. Some examples of fungi from this phyla are shelf fungi, toadstools, puffballs, and jelly fungi. This phylum reproduces sexually. First, it germinates to produce haploid mycelium, which begin to grow, one positive and one negative. They then fuse to form a secondary mycelium that contains haploid nuclei of each mating type. These grow in the soil for years and can grow up to hundreds of meters long. With the correct moisture and nutrients and correct environment, they sprout fruiting bodies with spores that push above the ground that we call mushrooms. When the mushroom cap opens, it exposes tiny gills on its underside, which are lined with basidium. The two nuclei in each basidium fuse to form a diploid zygote cell, which then undergoes meiosis and forms clusters of basidiospores that form at the end of each basidium. These basidiophores are then spread out, and that completes the sexual life cycle of Basidiomycota. Basidiomycota also form a fairy ring, and that's when the soil grows depleted over time, so the new mushrooms only sprout at the edges of mycelium, and that produces the fairy ring, which is why it's in a perfect circle.
The reproductive cycle of this common mode takes a lot of time and patience because its sole purpose in life is to make sure the genetics are passed on to the next generation. The first step starts out with hyphae. The hyphae fuse with the opposite mating type, forming a gametangia. The gametangia then produces gametes. Opposite mating types of the gametes then fuse together to form a zygospore. The zygospore contains the zygote, which remains dormant for a long period of time. Once the zygote becomes active, it produces a haploid spore. As you can see, the mold is slowly taking over the bread by using its rhizomes to release digestive enzymes to absorb the bread.
The second phyla of fungi is called Ascomycota. What is Ascomycota? It is also known as a suck fungi and is the largest phylum in the kingdom with over 30,000 species. Some are visible like morels, cup fungi, and others are microscopic like yeast. There are individual cells in the ascomycetes as they have crosswalls in their hyphae. Cytoplasm and nuclei moves in the tiny holes in their cross walls. Ascomycetes are heterotrophic as they cannot make food on their own, which means they need help from others. Where can we find ascomycetes? They are well adapted to their surroundings and almost all are parasitic or terrestrial. A few are adapted to freshwater and marine environments while others have the ability to grow underneath and non-living things in their environment. Ascomycetes also use the camouflage technique like color, size, and texture. How do ascomycetes reproduce? They can reproduce both sexually or asexually. In asexual reproduction, tiny spores called cornidia are formed at the tips of specific hyphae called cornidiopores, which means dust. Asexual reproduction occurs when a cornidium, the singular word for cornidia, falls in a suitable environment, which then grows in a haploid mycelium. Ascomycetes can also reproduce sexually. Step 1. Haploid hyphae of two different mating types, a positive and a negative, come close together. Step 2. The N plus N hyphae then creates a fruiting body in which sexual reproduction proceeds. Step 3. Fusion occurs and gametandria from the two mating sites fuse, but the haploid nuclei don't fuse. Step 4. Fusion produces a hyphae N plus N that has a haploid nuclei from each of the mating types. Step 5. Within the fruiting body, the ascus forms. Step 6. Inside the ascus, two nuclei of different mating types fuse to form a diploid zygote. Step 7. Zygote divides by meiosis, producing 4 haploid cells. Step 8. Meiosis then is usually followed by a cycle of mitosis, producing 8 cells called ascospores. In this short demo, we took 1 teaspoon of yeast powder and added 2 teaspoons of sugar to a small amount of warm water, and in about 18 to 20 minutes, we were able to see cell division happening in rapidly growing yeast cells. And if we were to put this under a microscope, the cell division would look like this.
Dectomycota includes many fungi that are not able to be placed in other phyla because they don't include a sexual phase. Since fungi mainly are classified by their reproduction phase, the ones that don't have a phase are placed in Dectomycota altogether. It's known as imperfect fungi because biologists can't figure out what to do with them. Many of the fungi resemble Ascomyceta. The other ones are closer to Basidiomyceta, and also some can be pretty close to Zygomyceta. Do you know the most well-known type of Dectomycota? Well, it's Penicillium notatum. It grows on many fruit and is also the source for the vaccine used greatly in the 20th century, which is called penicillin.